So like he said, in my group, we're developing treatments for aggressive cancers, in particular glioblastoma and triple-negative breast cancer. Glioblastoma is an aggressive form of brain tumor. Median survival for this disease is 12 to 15 months. 5-year survival is less than 10%. And for patients who do survive, there's often very dismal side effects because treatments often hit the normal brain in addition to the tumor. Since I moved to Delaware, I got very interested in triple-negative breast cancer because Delaware is actually the state that has the highest incidence of that disease. So it accounts for almost a quarter of all breast cancers, and unfortunately, it's unsusceptible to conventional targeted or hormonal therapies because it doesn't express the right receptors. So for targeted therapies, you're looking at HER2 receptor, progesterone receptor, estrogen receptor, and triple-negative breast cancer lacks expression of those three. That's why it's called triple-negative. So for both of these, there's really a desperate need for new treatments. They're currently treated with surgery, radiation, and chemotherapy, which is the same thing used to treat most forms of cancer. Unfortunately, these are very invasive. They have nonspecific toxicity and leave behind residual disease. So really, we think there's a pretty substantial need for new treatments that enable high precision and very effective treatment. And we think that nanoparticles in particular can help us meet this need. The reason we like to use nanoparticles is that they're tailorable tools, as we kind of just saw in the last talk, for nanomedicine. So what's great about nanoparticles is that they're small enough that they can get into tumors by exploiting the leaky vasculature, but they're big enough that we can give them very unique functionalities, and that's something you often can't do with molecular level systems. And sometimes we can impart multiple functions within a single nanoparticle as well. And because of this, we can have new ways to treat disease that are very specific and very effective. Now, in my group, we have several different technologies that we're developing to try to enable high precision therapy. And I'm not going to go into all of them today, but I did just want to give like the one slide overview of what my lab group does. So some of our work is in gene regulation, which I will talk about quite a bit today. We also work in photothermal therapy, and I'll talk about that a little bit. The things I won't get to talk about are drug delivery, and we also work in making biomimetic particles, so particles made to look like cells so that they can sort of hide from the immune system and get where they need to go. And then we also do some work in light-triggered release of payloads. So these three on the left are what I'll focus on today, starting off with the photothermal therapy, which is actually a nice segue from the end of the last talk. So as just introduced, photothermal therapy is a treatment where you deliver nanoparticles into a tumor, and then you can irradiate with near-infrared light, which will penetrate through overlying tissue to get to your particle-laden tumor below. And if the particles have the right optical properties, they can then convert that light energy into heat to physically damage the cancer cells. I've been working on photothermal therapy for over a decade now, which is hard to believe. And these are just some of the papers that I've published in this area. The ones from my group, since I started my lab at Delaware, are highlighted in blue. And the main type of nanoparticle that my group works with is nanoshells, which are silica core gold shell particles, as drawn in this cartoon here. We've shown in the past that these particles can be systemically delivered to subcutaneous tumors. So this is a subcutaneous brain tumor, where here what you're seeing is particles in red, the tumor cell nuclei in blue, and the tumor vessels in green. So you really see they're exploiting the vascular network to get where they need to go inside the tumor. And once we apply the laser, we see that this treatment is very effective. So on the left here, we have representative animals where you see for a nanoshell-treated mice at day 0, before the laser is applied, it has a huge tumor, but by day 7, that tumor is completely gone. If you look at a saline-treated mouse, its tumor just continues to grow. So if you then look at the aggregate data for all the animals in the study, you see that none of the mice treated with saline lived beyond 24 days, whereas 4 of 7 mice in the nanoshell plus laser treated group displayed complete tumor regression. And that was really exciting because, as I mentioned before, these tumors are very aggressive. So this was just a simple subcutaneous tumor model. We did then move on and do a more sort of physiologically relevant model where the tumors were actually implanted in the brains of the mice. I'm not going to go into that in detail, but if you're interested in that work, that was published in Nanomedicine back in 2012. Now, in my group, one of the things we're actively pursuing is looking at combining photothermal therapy with other therapies, with the idea being we can have sort of a more comprehensive cancer treatment. One of the things we did recently is basically use photothermal therapy to promote drug entry into cancer cells. Here, the idea was that if you have your cancer cells with the nanoshells and then drugs are in the vicinity of the cells, once you apply near-infrared light, it will cause membrane damage to the cells, and that will allow more drugs to shuttle into the cell and be more effective. So this is the cartoon version of what that paper showed. I'm not going to go into the details, but if you're interested, there's the reference. Another thing that we're currently developing is combining photothermal therapy with photodynamic therapy. So photodynamic therapy is a different type of light-activated therapy where you have molecules called photosensitizers that transfer absorbed light energy to adjacent tissue oxygen molecules, and they basically convert those to toxic singlet oxygen that'll do things like induce DNA damage. My collaborator, Joel Rosenthal, in chemistry at Delaware, has made a really effective photosensitizer. The paper describing that work is now in revision and should hopefully be online soon. And the next step that Joel and I are pursuing is taking his photosensitizer and conjugating it to the nanoshells so that we can have a single platform enabling both photodynamic and photothermal therapy in a single entity. One thing I do want to talk about in a little bit more detail, again kind of building on the last talk actually, is developing a biodegradable platform for photothermal therapy. So this is just a slide sort of showing some of the different types of nanoparticles used for photothermal therapy, and you see most of them are gold. So we have nanoshells that my group uses, nanorods, nanocages, nanostars, nanoschnozzberries, right? There's many different types of gold-based particles, but as mentioned before, these are going to stay in the body for a very long time. So my group, and Danielle Valcourt in particular, is working on creating polymer-based particles that have near-infrared dyes encapsulated inside so that we have a biodegradable platform for photothermal therapy. So quickly, I just wanted to go through some of the work that she's done. In essence, she takes polylactic coglycolic acid, mixes it with infrared 820 dye, IR820 dye, and acetone. That gets added dropwise to water. You then stir, purify, and characterize these by a number of techniques. And what you see here is that these particles have a size of about 60 nanometers in diameter, and their surface charge is about negative 40 millivolts, and we see they do in fact encapsulate that IR820 dye, as shown by the high absorbance peak out at 800 nanometers. One of the things we really wanted to confirm was that encapsulating this dye doesn't hinder its ability to produce heat. So we simply took vials filled with either free dye or encapsulated dye and used a thermal camera while we were irradiating with a laser to look at the heating profile. And you see basically that the free dye and the encapsulated dye show similar heating profiles. One of the other things we wanted to look at was stability of the system. So one of the limitations of freely delivered dye is that it's not very stable. So you see at day 0, this is the absorbance peak for the free dye, but by day 30, it's really dramatically decreased, meaning it won't be as effective as a heating agent. But when we have the dye encapsulated in the particles, it remains stable over a 30-day period. So this is one major advantage of encapsulating this dye inside of a nanoparticle. The other major advantage comes in terms of cytocompatibility. So this is looking at relative metabolic activity of cells that are exposed to the free dye. Here, there's no light exposure. This is just the dark toxicity of the dye. So you see above about 15 micromolar, it starts to drop off. But when you encapsulate the dye in the particles, it's cytocompatible across all the doses tested. So again, by encapsulating the dye, it's more stable and it's more cytocompatible. So we've now started to evaluate the potency of these particles simply by adding them to cells and then applying a laser. And what you see here is that saline-treated cells that are exposed to no laser or with laser are totally viable. The particle-treated cells that get the laser treatment have a dramatic drop in metabolic activity. So overall, what this tells us is that these particles can encapsulate the dye and are able to induce photothermal therapy. So just to quickly summarize photothermal therapy, I think it's sort of the quintessential example of a high-precision therapy because you're only ever going to have treatment where particles and laser are combined, right? And you can very precisely tailor where the laser will go. You can use it to enhance the effect of other treatments, and you can use both gold-based and polymeric particles to facilitate treatment. We do have our first in vivo study with this IR820 formulation ongoing, so hopefully next time you guys see me talk, I can give some of the in vivo results. All right, the next thing I want to talk about is our work in gene regulation. So I think of gene regulation as a high precision therapy because the idea is that you want to target genes that are expressed just in the cancer and not in the rest of the body. And in particular, we try to regulate gene expression with small interfering ribonucleic acids, or siRNA. The way this works is that you deliver double-stranded RNA into the cell. It binds the RNA-induced silencing complex called RISC. The two strands unwind, and the strand that remains bound to RISC guides it to perfectly complementary messenger RNA molecules in the cell cytoplasm. Those mRNA molecules are then degraded. And since mRNA normally encodes for protein, basically by causing this cleavage, you no longer get production of the disease-encoding protein that siRNA was targeting. Now, unfortunately, this works really well in a cartoon and not so great in reality. There's a lot of barriers to clinical application of nucleic acids. So they are rapidly degraded by nucleases, so they often don't get to the target tissue if you were to try to deliver them on their own. And even if they did get in the vicinity of the right cell, it's never going to just passively cross the cell wall like is shown in this cartoon. So RNA is very negatively charged. Cell membrane is negatively charged. They don't want to interact, right? So you really do need novel carriers to enable this delivery and ultimately translatable RNA interference. Now, one of the carriers that's been studied extensively in the literature is a molecule called polyethyleneamine. This is a really, really branched polymer structure that contains a lot of amine groups. As a result, it's very positively charged, and that allows it to complex with negatively charged nucleic acids. And what you get is essentially these randomly assembled polyplexes. So there are several advantages of PEI in gene regulation. It gives you very robust nucleic acid delivery capabilities. It's got long-term stability at 4 degrees C. It's inexpensive and widely available. You can easily chemically modify it. However, it is wildly toxic, which is not so great for clinical use. So one of the questions my group had is, can we somehow reduce the toxicity of this molecule by changing the way it is presented to cells? And more particularly, we wanted to know if we could exploit spherical nucleic acids as a way to change the architecture, enhance PEI-based siRNA delivery. So in the intro, it was mentioned that I worked with spherical nucleic acids in my postdoctoral fellowship. These are nanoparticles that consist of gold that is very densely coated with highly oriented RNA molecules, shown here in green on the surface, as well as polyethylene glycol for stability. So you see these really have this unique sort of core shell structure of the gold and the nucleic acid shell. Compared to linear nucleic acids, these particles have a lot of really unique properties. They offer, as I just said, very controlled RNA architecture. They get into cells pretty well. They are more resistant to nucleases, and they have negligible immunogenicity. So what we decided to ask in my group, this is my student Jillian Melamed who did this work, is can we use PEI-wrapped SNAs as a structure that has very precise, sort of precisely defined presentation of both the RNA and the PEI? If we make this structure, can it outperform these PEI polyplexes that just have random assembly of the PEI and the RNA? To answer this question, Jillian made both of these constructs and then looked at their effect in an in vitro model of glioblastoma. And in particular, she looked at their cellular uptake, their ability to escape lysosomal compartments and get into the cytosol. This is particularly important because RNA has to reach the cytosol to be effective because that's where that RISC complex is located. And then, if they are able to get out into the cytosol, what is their gene regulation capability and how cytocompatible are they? To make these particles, Jillian took bare gold particles, coated them with thiolated RNA and thiolated PEG. So this is your base bare spherical nucleic acid construct. and then she electrostatically adsorbed branch PEI to the surface. These are just some TEM images showing the overall structure of these. So you see that both the SNAs and the PEI SNAs have this sort of white nucleic acid shell surrounding the gold nanoparticle core. To make the polyplexes, she simply mixed the RNA and the branch PEI together, shake, 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 and you're done. So it's a little bit of a simpler process. Looking at some of the characteristics of these particles, she saw that for the PEI SNA system, you get about 40 RNA duplexes per particle, and you get close to 1,000 PEI molecules per nanoparticle. And then over here on the right, we've got analysis of their charge as well as their size. So Barragold particles and spherical nucleic acids are negatively charged, but once you add PEI to those, you start to have a positive charge, similar to that of polyplexes. Likewise, if you look at the red dots now, this is size. You see that gold particles, the size goes up slightly for SNAs. Once you add the PEI, it goes up to about 150 nanometers, which is similar to that of the polyplexes. So in this study, it was important that the PEI SNAs and the polyplexes had similar size and surface charge because we didn't want that to be a factor in the analysis. We wanted really just to be looking at the role of architecture and not these other characteristics. All right, so the first things Jillian looked at were cell uptake and cytosolic delivery. So to look at cell uptake, she did some flow cytometry analysis, where here the particles are labeled with fluorophore labeled RNA, so Cy5 labeled RNA. And I'll just focus in on the 24-hour time point shown at right. And what you see is that, so PEI SNAs here are blue. So you see the most uptake for PEI SNAs, the next most for the polyplexes, the third most for the SNAs. So what this tells us is that the extent and the rate of the particle internalization really depends on surface chemistry and architecture, because we know that the PEI SNAs and polyplexes had similar charge. They were both positively charged, and they get into cells better than the negatively charged SNAs. But for some reason, the PEI SNAs are showing a dramatic improvement even over the polyplexes. So that means it's not just the chemistry, but also the architecture. This is just some charts showing the kinetics for each individual particle type. So you see SNAs pretty much stabilize their uptake around 4 hours, whereas polyplexes and PEI SNAs show continuous uptake through 24 hours. Now, if you look inside of cells at where these particles are going, we also find that lysosomal accumulation depends on nanocarrier architecture. In the top row, we have polyplexes, where again the RNA is labeled with Cy5. Lysosomes are labeled with LAMP1 MGFP, and over here you have a merge. So for polyplexes, we see in overlap, so-called a Manders colocalization coefficient, this MCC, of 0.84. But for the PEI SNAs, we see a decrease in the overlap between the RNA and the lysosomes. So it goes down by almost 20%, down to 0.67. So what this tells us is that lesser in lysosomes, indicating more are getting out into the cytosol. And again, this is really important in order for RNA to be effective. So then what we wanted to know is that extra amount that's getting out of the lysosomes having an influence on gene regulation potency? To assess this, Jillian used a model where we delivered siRNA targeting green fluorescent protein. So we had cells that expressed GFP. And you see with the polyplexes delivering 50 nanomolar of RNA, there's no gene silencing observed. But when she used the PEI SNAs delivering 40 nanomolar of the RNA, she did see silencing of GFP expression. And in order to get a similar effect with the polyplexes, she had to go up to 500 nanomolar of siRNA. So she did a sort of dosing experiment in order to calculate the effective dose for 50% silencing, so the ED50 for each of these systems. And you see for the polyplexes, it was about 400 nanomolar, but for the PEI SNAs, it was 40 nanomolar, telling us the particles are 10 times more effective than the polyplexes. So finally, what about cytocompatibility, right? I told you the biggest limitation with PEI is that it's too toxic. So to assess this, Jillian looked at the relative metabolic activity of cells exposed to free PEI in light gray bars or polyplexes in the dark gray bars. And what you see is that above about 10 micrograms per mil, super duper toxic to the cells, right? They're basically dead. But when she delivered the same content of PEI in the particle form, we see 100% viability of the cells across all of these doses. So this, coupled with the last sort of data on gene regulation, was what got me really, really excited. Just showing how much of an influence architecture can really have on both potency as well as safety. So overall, we've seen that these PEI SNAs outperform polyplexes as gene regulatory agents because they improve cell uptake, lysosomal evasion, and cytocompatibility. And I thought this was a pretty fun project in that it showed that these spherical nucleic acids could serve as a platform to control architecture and sort of answer some fundamental questions about, these hybrid nucleic acid polycation type systems for RNA delivery. A story that I'm not going to go into. Oh, I should also highlight, this was actually just published last week in Molecular Therapy Nucleic Acids, so read it. All right, so I don't have time to go into it, but in my group, once we started getting some of the data on that last system, we got interested in whether this idea that architecture matters would extend to other platforms as well. And so we actually published another paper last week in Cellular and Molecular Bioengineering that shows this is the case. So we made this system that consists of nanoshells coated with alternating layers of polyl-lysine, microRNA, which is similar to siRNA, but instead of targeting a single gene, it regulates multiple genes simultaneously. So you have polyl-lysine, microRNA, and then polyl-lysine in this layered structure. And we compared those to polyplexes, so these randomly assembled PLL microRNA systems. And again, we found that the particle outperformed the randomly assembled polyplex. So regardless of whether it's a PEI SNA or this layer-by-layer type system, sort of my take-home for this part is that architecture is really, really important. All right, so I told you architecture matters, and we can do a lot of great things with these hybrid polycation nucleic acid systems. One of their limitations, though, is that technically they would regulate gene expression in any cell that they entered, right? They don't have any way to offer the specificity that we might really, really want in a cancer application. So one of the things my student Rachel Riley has been working on is developing nanoparticles for on-demand gene regulation. The idea here is that if we take nanoshells that are coated with siRNA, once we apply a pulse laser, we can break the gold thiol bond that's linking that RNA to the particle surface. That released RNA can then go bind the RNA-induced silencing complex to silence the target gene of interest, which in our work was GFP. So to evaluate this sort of idea, Rachel made nanoshells coated with RNA that had Cy5 fluorophores on their exterior. She put them in a cuvette with a little stir bar. So the sample is sort of stirring while she's irradiating with a pulse laser. She then took that sample, centrifuged it to form a pellet of the nanoparticles, and any released RNA she took off in the supernatant. So the released RNA she characterized by a number of techniques, and likewise she characterized the particle pellet by a number of techniques. So the first thing she did was an olivgreen assay just to quantify the amount of RNA that was released from the particles. And what you see here on the left is that as the irradiation time goes up, the amount of RNA coming off the particle goes up. So we see that with a 30-minute exposure, we get about 75% or so release of the RNA. If you then look at the particle pellet and the size of those particles, you see that as irradiation time goes up, the particle size goes down. And this makes sense because if the RNA is coming off, the overall size is going to shrink. So with this 30-minute exposure, we're now down to a size of 150 nanometers, which is actually the size of our bare nanoparticles, again, showing most of the RNA has come off the surface. One of the questions we had was, is the released RNA primarily double or single-stranded? So one of the possibilities is that instead of breaking the gold thiol bond, you might actually just cause duplex denaturation where they separate from each other. And we want primarily double-strand release because double-stranded RNA is what's recognized by risk to enable gene silencing. So what Rachel did is she took that released RNA and ran it on a gel so that she could look at the structure. And what you see here, oh, it's really dim on the slide. Sorry about that. if you go to the paper, it's in nano letters, you'll see it much clearer. So this is basically the samples that were exposed to the light here. And I promise in the paper you see there's really nice bright bands here at the duplex region, but not so much at the antisense strand region. So what this tells us is that we are triggering release of primarily duplexes, not single strands. The next question, though, was, is the released RNA still functional, or is it somehow damaged in the release process? To answer this question, she took that released RNA and just transfected it into cells with a commercial transfection agent. And what she found is that it is still functional. You still see reduction in GFP expression. This is just representative data on the left and aggregate data on the right. So taking all of this together, we finally said, okay, can we use these to enable on-demand gene silencing? Rachel cultured cells, added our nanoshells coated with siRNA targeting GFP or a scrambled RNA, visualized their uptake in the cells by microscopy and also quantified it by flow cytometry, and then irradiated samples to trigger the RNA release and analyze GFP expression in the cells by flow cytometry. And what you see here is that the particles do enter these GFP expressing cells. So the particles here are red, the cells are green and blue, and you see that quantitatively as well by flow cytometry. There's a very high Cy5 signal indicating the RNA inside of these cells. Once we then apply the laser, what you see is very robust silencing of that GFP expression. So here, if you just look, this is representative data on the left and aggregate data on the right. You see it's only when we have the siGFP nanoshells with laser, so this first group, that we have silencing of GFP expression. In all of the controls, there was no silencing of GFP. So overall, these particles are really unique in that they can enable on-demand gene silencing. So you can choose when to trigger your laser, where to trigger your laser. So that's a really unique advantage of the system because, as the second bullet point says, if you trigger your RNA release only when and where you want it, you can imagine that you would have a very high therapeutic ratio because your off-target effects would be very minimal. All right, the last thing I want to talk about, so the previous section was talking about how do we regulate signaling from inside the cell. That is very hard to do. So one of the things Rachel has also been interested in is developing ways to regulate signaling from outside the cell. So most cancer cells express receptors on their surface. So that's what this little yellow thing is supposed to be, is a receptor on the surface of a cancer cell. These receptors will interact with ligands in the extracellular environment to activate some sort of signaling pathway that makes the cancer do all of its aggressive things. Antibodies are used therapeutically to bind these receptors so that the ligands can't bind, and that reduces the signaling pathway. And the question that Rachel and I had was, could we use antibody-nanoparticle conjugates to get an even more effective inhibition of signaling? The idea here is that antibody-nanoparticle conjugates could bind multiple receptors simultaneously. This is something called multivalent binding. And if we're able to do that, we could have basically a more robust suppression of signaling than you get with just freely delivered antibodies. For treating triple-negative breast cancer, we thought the Wnt signaling pathway would be a very good therapeutic target. This is a simplified diagram of the Wnt signaling pathway in a normal cell. And basically, the only thing you need to know in normal cells is that there's this destruction complex that constantly degrades beta-catenin. Beta-catenin is sort of the key mediator of Wnt signaling. So when beta-catenin is degraded, Wnt target genes are off. Now, in cancer cells, there's an overexpression of these frizzled receptors. They bind Wnt ligands in the extracellular environment. So Wnt ligands bind. This inhibits that beta-catenin destruction complex. So beta-catenin is no longer degraded. What happens then is it accumulates in the cytoplasm, enters the nucleus, and turns on Wnt target genes. And these genes do things like control proliferation, drug resistance, migration. So ultimately, you have tumors that are more aggressive, more prone to metastasis, things like that. So our hypothesis was that if we could create nanoshells to bind these frizzled receptors, we could block the Wnt frizzled interactions to basically shut down the signaling pathway and do so more effectively than freely delivered antibodies. To create these particles, Rachel started with nanoshells and coated them with antibodies using a heterobifunctional PEG linker. She also backfilled with PEG for stability. Here you see that compared to bare particles, the antibody-coated particles are somewhat larger, and they also have a more neutral zeta potential. With these, we have about 50 antibodies per nanoparticle. We tested these in a couple different cell culture models. So MDA MB231s and BT20s are two different types of triple-negative breast cancer cell lines. MCF10As are a normal breast cell line. And what you see by both the immunohistochemistry and the Western blot is that the cancer cells dramatically overexpress frizzled compared to the normal cell line. The first thing Rachel wanted to look at was how strongly do these particles actually bind the targeted cells. So to assess this, she cultured the cells, exposed them to the particles or freely delivered antibodies, as well as some control nanoparticles that shouldn't bind. Imaged binding by multiphoton microscopy. And then to quantify the sort of strength of the binding, she added secondary antibodies conjugated to horseradish peroxidase. The idea is that these would bind antibodies on particles that are attached to the cell or antibodies on the free antibodies that are attached to the cell. We could then add a color-changing substrate, and basically the amount of color we get indicates the strength of the binding. Right? So first, just looking at the visualization of binding, here if you look at the MDA MB231s on the left, this is the triple-negative breast cancer cell line. You see that the frizzled 7 targeted nanoshells showed a lot of binding to these cells, but not so much to the healthy cells. And our control particles coated with just PEG don't really bind either cell type. Now, if we compare the targeted particles to free antibodies and looking at the binding strength, what you see here is a calculated dissociation constant of 4.8 times 10 to the minus 10th for the frizzled 7 nanoshells, 1.3 times 10 to the minus 8th for the free antibodies. So you can think of these dissociation constants as sort of the inverse of binding strength, binding avidity. So what that tells us is that the particles are binding with 100 times more strength than the free antibodies. But is that level of binding sufficient to block Wnt signaling? That was the next question we had. To answer this, cultured cells exposed to our particles or free antibodies. We also co-treated with Wnt3A in order to competitively bind those frizzled receptors, and then analyzed for expression of beta-catenin, which is that key mediator of Wnt signaling, as well as for axin 2. So axin 2 is one of the genes that is downstream of beta-catenin, and it's considered the definitive marker of whether Wnt signaling is on or off. What you see in these images is that these particles very dramatically shut down beta-catenin expression. So here we have control cells shown at the left, so cell nuclei are blue, beta-catenin is green. And you see with free antibody, there's really no suppression of beta-catenin expression, and same thing with our control particles. But when we have the frizzled 7 nanoshells, there's major downregulation of beta-catenin. And this was really, really exciting because with these particles, we actually delivered 50 times less antibody than was delivered with the free antibody. Likewise, when we look at relative axin 2 expression, you see it's only with the frizzled 7 nanoshells that we see decreases in axin 2 expression. Didn't see that effect in any of the other groups. And again, we delivered 50 times less antibody with the particles than was delivered in the free form. Functionally, we wanted to know what the effects of inhibiting Wnt signaling are. So just looking at metabolic activity of cells, you see that it's reduced when you deliver these frizzled 7 nanoshells. But looking in vivo, we were interested in what is the effect of these particles on lung metastasis. So most breast cancers will metastasize to the bone. Triple-negative breast cancer is unique in that it tends to metastasize to lung. And that is driven largely by this Wnt signaling pathway. So we wanted to know, just in a pilot study, this is very preliminary data I'll be showing you, do these particles have potential to combat this lung metastatic TNBC? So in this model, you deliver MDA MV231 cells via the tail vein and wait a few weeks, and you'll start to get metastases forming in the lung. And if you engineer your cells to express luciferase, you can actually visualize that lung metastasis formation. And once you excise those tumors, you can sort of stain and confirm that they do express frizzled 7. That's what you're seeing here in the brown color. So the first thing we were interested in was, do these particles even go to the lung? Right? They can't be a good treatment if they don't get to the organ of interest. So what we did is followed a treatment schedule here where we injected cells, injected particles on the days shown here, and then at the end of the study, excised the tissues for analysis of gold content by ICP mass spec. And I should note that this is a log scale, so we see pretty minimal accumulation in blood, brain, femur, heart, intestine, and kidney. We do see high accumulation in the liver and the spleen, which are the clearance organs, right? So that's not very surprising. But what got us super excited is that we actually see a lot of accumulation in the lung. So that was really, really exciting to see. And if you look a little more closely at the lung, this is a dark field image where the tumor is sort of on the left half. So you see where the tumor sort of barrier is. So tumor's over here on the left. The adjacent normal lung is on the right. And the particles show up as this kind of golden, orangey, reddish color. And we saw that the particles seem to be preferentially binding to the tumor and not the normal lung. So they're in the lung, and they're in the right place within the lung. So that was super exciting to see. And then finally, if we just look at effect on tumor growth, it's hard to see the actual mouse in these images, but there is a mouse here. So if you look at just the luminescent signal coming from the tumors in the lungs, you see for a saline-treated mouse, it grows pretty dramatically over time. For this particular mouse, this was one of our better responders. You see that it has stable disease or potentially even regression of its lung metastases. And this is the aggregate data for all the mice in the pilot study. So saline-treated mice are black. The frizzled 7 nanoshell-treated mice are red. And at all time points, so these are towards the later end of the study, at all time points we see that the mean luminescence, so the mean size of these lung mets in the particle-treated group is lower than the saline-treated group. So this was a small-scale study. It was 3 to 4 mice per group. But this is the pilot data that I'm now putting in my grant to NIH to convince them to give me money to do a larger study to assess this in more detail. So overall, I've shown you that these particles can bind triple-negative breast cancer cells to selectively inhibit Wnt signaling and decrease cell viability. It does look like they have potential to enter lung metastases to reduce their growth. And the part that I think is super exciting is that by taking antibodies and putting them on the surface of a nanoparticle, we're able to increase their effect relative to freely delivered antibodies, and we believe that's due to multivalent binding effects. So overall, I've shown you that there are many different ways to attack aggressive cancers with high precision. So in my lab, we've developed particles that enable photothermal therapy, particles that enable gene regulation, and particles that enable signal cascade interference. And overall, I think my biggest take home that I hope you're learning from my talk is that nanoscale architecture is critical. So when we take RNA and put it on a particle in a three-dimensional form, it gets very unique properties that wouldn't exist otherwise. And same idea here with the antibodies. When we put it on a particle, it begins to be much more effective than the freely delivered agent. So nanoscale architecture is very, very, very critical. With that, I'm going to just acknowledge my group and my funding sources and thank all of you for your attention. Yeah, so the question is why with the PEI-wrapped SNAs compared to the polyplexes do we see reduced toxicity? The honest answer is I don't fully know yet. We have a running hypothesis that it has to do with the way the amine groups are presented to the cell, like maybe something about their structure is changing, but we're doing studies now to try to probe that in more detail so that we can give a very precise answer. But that's our sort of running hypothesis. One of the things we do know is that it doesn't seem like the RNA and the let me go forward, actually. The architecture does not change the way the RNA and the PEI traffic in the cells. So it seems like this is here looking at RNA overlaid with PEI. And basically, we see that they're co-localized regardless of whether we have it in the polyplex form or the particle form. So we don't think the difference in toxicity is related to trafficking of those. They seem to traffic together regardless of architecture. So we know that's not it, but we're still trying to probe in more detail. If they come off from the surface, will they have similar? Yeah.
Do you know if they came off or not?
Yeah. Based on this, I don't know whether they are all coming off the particle and moving together through the cell, or if it's still on the particle inside the cell. So unfortunately, kind of same answer as the last one. We don't fully know yet how these are unpackaging inside the cell, but something we're actively pursuing. Yeah. I mean, one reason is my group is an expert with nanoshells. We know how to make them. No, one of the reasons we really like nanoshells is they're already in multiple FDA clinical trials. So if they end up getting approved, we're already working with a template that's well on its way. They also have a lot of nice properties that we can exploit for imaging, which allows us to track them, which I think is a nice advantage as well. Yeah. Yeah. Yeah, so the question is about photodynamic therapy and light penetration. Let me see if I can scroll there while I'm talking. So the photosensitizer that Joel Rosenthal's group has developed absorbs 500 nanometer light, which will not penetrate tissue very well. So my cartoon showed it, which is why I'm trying to get back to it. Oh, there it is. All right, so this photosensitizer, it's called palladium dimethylbiladiene. It's a name for you. So it absorbs 500 nanometer light, won't penetrate very well. One of the things that these nanoshells can do, which I didn't mention before, is when we irradiate them with pulsed 800 nanometer light, in addition to producing heat, they can also emit light in the range of 400 to 600 nanometers. So our idea is that that emitted light can then activate the photosensitizer that's close to the particle surface. So that'll give us a little bit of a benefit in that we can now use near-infrared light, but even then near-infrared light will still only penetrate so far, right? So we would be looking mostly at applications where things would be readily accessible, so oral cancer, breast cancer, prostate cancer, things of that nature, or applications where you might be able to use a fiber optic probe to deliver the light. Yeah, technically, in this paper that's in revision right now, we have shown that the photosensitizer can be effective either inside or outside the cell, but it is more effective if we give it enough time to get into the cell.
Good. Thank you. In the interest of time, we'll close it up there and thank our speaker one more time.